It's a great pleasure to give this presentation today. It's really fun to see so many people here attending. So my lab really has two major avenues of research. One deals with how the nervous system is generated, which is something we've been studying for about 20 years. And this is something more novel, how the nervous system degenerates, how it breaks down, which is something we've only been working on for the last 2-3 years or so. So it's something that's quite novel in the lab. And this is I think my second lecture presentation talking about this work, so I'm not really that familiar with the subject myself sometimes. So what I was going to do today is to first give you a very brief introduction to what Alzheimer's disease is, and more from a very molecular point of view. Since I'm not a clinician, I'm not really on top of all the clinical manifestations of Alzheimer's and how it actually presents itself in patients, but I'll give you some molecular information. I want to give you a brief introduction to how the fruit fly Drosophila works as a model system and how it can be used as a tool for disease research. I will then talk about Drosophila as a specific tool for Alzheimer's research. I will give you a very brief overview of our work, and I'll talk a little bit one slide about some future plans we have. So first, AD. As you know, it's a very common disease. I think in Sweden there's some 100,000 people with Alzheimer's disease, and it's one of the really fast-growing diseases in the Western world, and will become very big in the developing world too as people become older. It's typically manifested in old age, and what you will see when people die from very severe Alzheimer's is that the brain is showing severe signs of degeneration. So cells have parts of the whole brain has actually been lost by cell death. So the cells are dying in there, and it creates degeneration of the whole structure. So you can see severe degeneration of the brain.